Hi, hi. So, today we have the last lecture in this week and also the last lecture when we speak about some concrete pathway, yes? Next week you have some overview lectures about the regulation, for example, but no new pathways, yes? So today the last. Today we should speak about the metabolism of glycogen and about synthesis of fatty acids and about synthesis of triacylglycerols, yes? But at the beginning, I should tell you something about the storage compounds that we have in our body. In our body, we have two main storage compounds, triacylglycerols in adipocytes and then glycogen. Glycogen you can find in every cell, but for our body is important especially liver glycogen and muscle glycogen. How we have already heard, in our body there is no storage protein, yes? If you destroy some protein, the body misses its function, yes? So all proteins we need. Triacylglycerols are better energy storage. For in 1 gram of fat, you have 6 times more energy than in 1 gram of glycogen. For in 1 gram of glycogen, there is not only the saccharides, but also the water. For glycogen is polar compound. It means it likes water. By complete oxidation of 1 gram of fatty acid, you gain approximately 38 kilojoules. From 1 gram of saccharides or proteins, you get 17 kilojoules, yes? Fatty acids are more reduced than amino acids or than saccharides, yes? And the more reduced, the more energy inside. It means that if you have 70 kilos, you have so many kilojoules in triacylglycerols, only 10,000 kilojoules in proteins, 2,500 kilojoules in glycogen, and only 170 kilojoules in blood glucose, in plasma glucose. It means that with these two energy sources, you can live only 1 day. How many kilojoules we need per day? How many? 10,000, 8 to 10,000. Yes, normally 8,000, that's the 8,000 kilojoules, it means 2,000 kilocalories. Yes? So what's glycogen? What's its structure? That you should know.
So polymer of glucose.
Yes, so it's the polymer of glucose. What type of bonds you can find in glycogen?
1,4.
Alpha 1,4, alpha 1,6. And glycogen you can find only in animal cells, yes? What polysaccharide do we have in plants? Starch, yes? Their structure is similar. Also alpha 1 to 4, alpha 1 to 6 bonds, but glycogen in animal cells, starch in plant cells. In our body, you can find glycogen in all cells, yes? Some small amount is in each cell you can find, also in the brain, in kidneys, in erythrocytes, also some small amount. What is for us important is especially the liver glycogen, approximately 100 grams of glycogen we have in liver, and 500 grams of glycogen in skeletal muscle, yes? Important is that these two glycogens have absolutely different function. Liver glycogen can be used to maintain the glycemia, for in liver you have glucose 6-phosphatase. So it means liver can release glucose, pure glucose. In our skeletal muscles, there is no glucose 6-phosphatase. It means skeletal muscle cannot release pure glucose, and therefore the muscle glycogen is only for the internal use of the muscle cell, yes? rhabdomyocyte. So different function, the structure is absolutely the same, only muscle cells are missing one enzyme. This you have said correctly, so only from glucose, most residues are bound by 1 to 4 band, alpha 1 to 4 band. Here the branching is done by alpha 1 to 6 band, and then in these branches also only 1 to 4 band. In glycogen molecule you can find two ends, non-reducing ends and reducing end. Yes, the majority of ends are non-reducing ends, and only on these non-reducing ends you can do the reaction. Yes, you can make the chain longer or shorter, but only on non-reducing ends. The reducing end is bound to amino acid tyrosine. That's in a protein that's called glycogenin. It's the molecule where we can find the glycogen. Where in the cell? In mitochondria, endoplasmic reticulum, in cytosol, in membranes. Where? In cytosol is glycogen, and the glycogenin keeps the glycogen molecule in cytosol. Yes? Synthesis and degradation of glycogen. It's quite simple. Yes, both the synthesis and also the degradation. For we have only two types of bonds in the molecule. The synthesis, glycogen is in cytosol, and therefore also the synthesis is in cytosol. The first reaction you already know. When the glucose enters the cell, it is phosphorylated by two isoenzymes. So the first reaction is glucose phosphorylation to glucose 6-phosphate. In liver, catalyzed by glucokinase. In muscle, catalyzed by hexokinase. The second step is the isomerization of glucose 6-phosphate to glucose 1-phosphate. This enzyme you do not have to know. Third reaction, for you know that glucose 1-phosphate nor glucose 6-phosphate aren't macroergic compounds. Yes, the bond glucose to phosphate, that's the ester bond, and it's not macroergic. And for the synthesis of glycogen, you need to have the activated glucose. Yes, with the macroergic bond. Therefore, glucose 1-phosphate has to react with some macroergic compound, and here it is UTP. What does it mean, UTP? What is it? Yes, uridine triphosphate, so the same as adenine triphosphate, adenosine triphosphate. And now, so it's the analog of GTP, ATP, yes? And it forms the UDP glucose, yes? So here, where 4 phosphate atoms, here only 2, yes? So 2 phosphate atoms are released. UDP glucose, that's the activated glucose, or also we said the macroergic glucose, yes? Enzyme also you do not have to know. And the UDP is bound to the first carbon, yes? So the same as here. And now you have the glycogen, you have UDP glucose.
Yeah, are you basically saying that we don't need to know any of these enzymes? We don't need to know any of these enzymes?
You need to know this, and then this.
Yeah.
So not these two. This you have to know, for these enzymes are enzymes of glycolysis, so that you have to know. And then for glycogen synthesis, you have to know this. This enzyme forms the alpha 1 to 4 bond, and then you have to know the enzyme that forms alpha 1 to 6 bond. Yes, that's on the next slide. So the UDP glucose reacts with glycogen molecule with the non-reducing end of glycogen. The enzyme is called glycogen synthase. Synthase for no ATP you need, yes? The energy was added here, therefore it's synthase, yes? Directly in this reaction, no ATP, therefore synthase. So glucose and the UDP is released from the molecule, and it can be rephosphorylated to UTP and to start the new reaction. Glucose glycogen synthase can catalyze only alpha 1 to 4 reaction bonds, yes? But of course that in glycogen you have also alpha 1 to 6 bonds, yes, for the branching. To catalyze these bonds we need to have other enzyme, yes? And the enzyme is called simply, it's branching enzyme, yes? And the reaction is also simply, if you have the linear chain of the glycogen, you take some oligosaccharide between 6 to 7 glucose residues, and you take this oligosaccharide and put them on the 6 carbon and produce alpha 1 to 6 bond, yes? So the branching enzyme cuts off 6 or 7 glucose molecules and put them on the and forms a branch with alpha 1 to 6 bond, yes? For this, you do not need any activation and ATP, yes? And these branches then can be further prolonged by glycogen synthase, by alpha 1 to 4 bonds. So for the synthesis of glycogen, you need to know 2 enzymes, glycogen synthase, alpha 1 to 4 bond, and branching enzyme, alpha 1 to 6 bond. Yes? And then, of course, the hexokinase or glucokinase. What about regulation? The key regulatory enzyme is glycogen synthase. And the main regulatory factor is phosphorylation or dephosphorylation. And the phosphorylation is regulated by hormone action, by insulin, or by glucagon. Glycogen synthase catalyzes anabolic process, formation of big polysaccharide, and therefore it's activated by anabolic hormone, by insulin. And the counter-regulatory hormones, as glucagon, or adrenaline, or cortisol, inhibit this process. Yes? And they do their action through the phosphorylation or dephosphorylation. Yes? Insulin in the cell activates dephosphorylation. So insulin activates dephosphorylation, and therefore it activates glycogen synthase. Counter-regulatory hormones, on the other hand, activates phosphorylation, and therefore they inactivate glycogen synthase. How this works, why insulin activates phosphorylation, why insulin activates dephosphorylation and glucagon phosphorylation, we will speak next week, yes, during the lecture about regulation, how it works. Do you have any questions to this, to glycogen synthesis?
Can the enzymes both cut and create the bond?
Whether they can?
The glucagon synthase, can it both create the bond and split the bond?
Not. For the splitting, we have other enzymes. Now we will speak about it. Here we have them. So that was glycogen synthesis. Glycogen degradation. Glycogen degradation is also simple. It's localized in cytosol, where you have the glycogen. And for the glycogenolysis, you need 2 enzymes to destroy the alpha 1 to 4 bond. The first enzyme is called glycogen phosphorylase. Glycogen phosphorylase. This enzyme is able to destroy alpha 1 to 4 bond by the phosphorylated cleavage. Phosphorylated cleavage means that you take inorganic phosphate from the cytosol, and with this inorganic phosphate, you destroy the bond. Yes? Therefore, phosphorylated cleavage. Yes? Not hydrolytic, but phosphorylated cleavage. And in this reaction, you form not pure glucose, but you produce glucose 1-phosphate. Yes? So it's very efficient for you produce glucose with phosphate, and therefore you do not have to use energy in ATP to produce the phosphorylated glucose. Yes? This glucose 1-phosphate is also called choreester. Yes? The second step for you know that glucose 1-phosphate, it's not the center of the glucose metabolism. The center is glucose 6-phosphate. And therefore, there is isomerization of glucose 1-phosphate to glucose 6-phosphate. This enzyme you do not have to remember. You have to know this, glycogen phosphorylase. So, only 2 steps. Here can be the third step. In muscle cell, here it's end. Glucose 6-phosphate can be used in glycolysis or in pentose cycle. If you are in liver, the phosphate can be removed from the glucose 6-phosphate molecule, and you will gain pure glucose. To do this third reaction, you need glucose 6-phosphatase. More difficult is how to remove the alpha 1 to 6 bond. For the synthesis, you need only branching enzyme. Here, unfortunately, you need 2 enzymes. The problem is that glycogen phosphorylase cannot destroy bonds, or it stops at the 4th glucose before the branching point. Yes? So 4 residues before branching point, the glycogen phosphorylase stops. In the first reaction, you take 3 glucose residues from this 4 glucose chain and transfer them to the main chain. Yes? The enzyme you do not have to remember, but the first process is transfer of 3 glucose residues. Yes? So this enzyme destroys alpha 1 to 4 bond. Yes? First, the branching is still there. The 1 glucose residue that is bound with alpha 1 to 6 bond is taken by debranching enzyme. Yes? So debranching enzyme that you have to know is the enzyme that removes alpha 1 to 6 bond. Yes? But you need more enzymes, not only glycogen phosphorylase and debranching enzyme, you need also this enzyme between. And now you have only the linear glycogen chain, and that's the place for glycogen phosphorylase action. Do you have questions?
Can you draw it?
Draw? Should I draw it? You have chain. This is glucose. Where I have some color. Here is the different bond, yes? Alpha 1 to 6. The red, that's alpha 1 to 6. And here we have the branch. So, this you can cut by glycogen phosphorylase. This you can also cut by glycogen phosphorylase. And now we have 1, 2, 3, and 4. And here the glycogen phosphorylase has to end. Yes, it cannot. Now you take the gluconotransferase that removes these 3 molecules It means destroy only alpha 1 to 4 bond and moves them here. Yes, so now you have so one alpha 1 to 4 bond destroyed, here formed the alpha 1 to 4 bond. And here comes the debranching enzyme. The debranching enzyme this enzyme destroys the alpha 1 to 6 bond. So it means now you have long linear main chain and glycogen phosphorylase can continue. So now it's clear? Regulation is very simple. It's opposite to synthesis. So the key regulatory enzyme is glycogen phosphorylase, and this enzyme is activated phosphorylated, is active phosphorylated. Glycogen synthase was inactive phosphorylated. And therefore, the hormones have also reverse effect. Insulin as an anabolic hormone inhibits, and counter-regulatory hormones, they are catabolic hormones, so they activate the pathway. Yes? In the books you will read that these hormones cannot do it directly, that they need one enzyme before, and the enzyme is called phosphorylase kinase. Yes? This enzyme, this kinase, does the phosphorylation of glycogen phosphorylase. Yes? Glycogen synthase can be directly modulated by these hormones. Here you need one step in between. Yes? So phosphorylase kinase is the enzyme that catalyzes the phosphorylation of glycogen phosphorylase. Very often in the books, you will also read only about phosphorylase and not about glycogen phosphorylase. Yes? For there is only one important phosphorylase in the metabolism, and that's the glycogen phosphorylase. Yes? Therefore, very often we do not use glycogen phosphorylase, but only phosphorylase. So that was everything about metabolism of glycogen. Everything clear? So now the more difficult topic, that's the synthesis of fatty acids. Synthesis of fatty acid is very difficult process, or you know, some steps are easy, but some steps are difficult. The easy steps are those steps that are similar to beta oxidation. Yes? It means that are similar to Krebs cycle. Therefore, at the beginning, I will tell you what's the difference between synthesis and degradation of fatty acids. It means between synthesis of fatty acid and beta oxidation. These two processes are opposite. Yes? When you have in beta oxidation, oxidation, in synthesis you will have reduction. Synthesis of fatty acids is localized in cytosol. Beta oxidation was in mitochondria. This is the compartmentalization. Means that one process is in cytosol, one in mitochondria. Yes? Intermediates of fatty acid synthesis are bound to this protein. We call it acyl carrier protein, ACP. There's a special protein that is used to carry these intermediates. For beta oxidation, we have coenzyme A. Yes? This protein is much, much bigger. Enzymes of beta oxidation are free in matrix, yes, in mitochondrial matrix. Enzymes of fatty acid synthesis form a big multi-enzyme complex that have 2 subunits with 6 domains. And this multi-enzyme complex is called synthase of fatty acids. Yes? Synthase, why it needs no ATP. What was the end product of beta oxidation? Acetyl coenzyme A. Yes? Therefore, the substrate for synthesis of fatty acids should be acetyl coenzyme A. That's true, but it's not enough. Yes? For in acetyl coenzyme A, there is not enough energy. And therefore, the pure substrate is this, a malonyl coenzyme A. It means that in the first step of synthesis of fatty acids, we need to produce malonyl coenzyme A, activated substrate. In beta oxidation, we used 2 cofactors, NAD and FAD. Here we use different cofactor. We use NADPH. Where we can produce NADPH? In pentose cycle. And the last difference is with beta oxidation you can degrade almost all fatty acids. Only the very long chain fatty acids you cannot degrade. But the fatty acid synthase can produce only one fatty acid. It can produce only palmitate. It means 16 carbon, monocarboxylic acid. And all other reactions, it means elongation or formation of double bonds, are catalyzed by other enzymes. So here only one product, and to do the other fatty acids you need other enzymes. The first step of fatty acid synthesis is the production of malonyl coenzyme A. It means of the activated substrate. How it works is that you take acetyl coenzyme A. Do you know how it looks like malonyl coenzyme A? So how many carbon atoms has malonyl? 3. This one is bound to coenzyme A. So here is double bond. And what I should draw here? H2, yes. And carboxy group. Yes. So it's produced by carboxylation of acetyl coenzyme A. For the carboxylation, you need ATP. So here, you have the ATP, the energy. Yes? But this reaction is not catalyzed by the fatty acid synthase. It's catalyzed by other enzyme, but the free enzyme that's called acetyl coenzyme A carboxylase. Yes? So this enzyme is free in the cytosol. For all carboxylations, you need cofactor. The cofactor is biotin. It means vitamin H or vitamin B7. Yes? In some books, you have vitamin H. In other books, vitamin B7. This enzyme, you have to remember, acetyl coenzyme A carboxylase, for is the regulatory enzyme. Yes? First reaction, regulatory enzyme. And what's funny is that this malonyl coenzyme A is destroyed in the first reaction. For in the first reaction, you remove the carboxy, this from it. Yes? So this reaction is here only to add more energy in the molecule For the condensation reaction, for the first reaction on synthase of fatty acids, you need more energy. Not only energy in acetyl coenzyme A, but the energy that is malonyl coenzyme A. Yes? Only this reaction is not catalyzed on fatty acid synthase. So the fatty acid synthase is a very big multi-enzyme complex. Here you have the name of enzymes that it has. You do not have to remember them, yes? But you see that in this one enzyme, big molecule, you have many enzymes, yes? The mammalian fatty acid synthase consists of 2 identical subunits, yes? And one subunit has 3 domains, yes? You do not have to remember what is where, but only take in account that it's a very big and complex enzyme. And therefore, there can be many problems in this enzyme, yes? Some genetic problems. The intermediates of the fatty acid synthase can bind on 2 different places. The majority of reactions take place here on the acyl carrier protein. Yes? But for the first reaction, you need this place. There's the phyl group on cysteine. And this amino acid is in one enzyme that's called condensing enzyme. Yes? In Harper, there is very detailedly described how it works. Yes? You do not have to know it absolutely in detail, but only take into account that it's very complicated pathway at the beginning. Here we have the reactions. The first reaction you already know. Yes? There's the formation of malonyl coenzyme A by acetyl coenzyme A carboxylase. Then you have 2 reactions when the substrates are bound to the synthase of fatty acids. In the first reaction, you bind acetyl coenzyme A, and in the second reaction, you bind malonyl coenzyme A. Yes? And you see that both of them bind to different binding place. Yes? One to condensing enzyme, the other to the acyl carrier protein. The names you do not have to remember. What reaction you have to know is this reaction. That's the condensation. When these 2 compounds, you condensate together. So you take acetyl bound to condensing enzyme, malonyl bound to acyl carrier protein. You release the carbon dioxide and produce acetoacetyl bound to acyl carrier protein. Acetoacetyl is this. And here is bound to sulfur in acyl carrier protein. So we have to know the first reaction and then the second, catalyzed by the condensing enzyme. The other reactions are simple. Yes, they are opposite to beta oxidation. So in beta oxidation, we had oxidation. So now we have reduction. Then in beta oxidation was addition of water. Here we have elimination of water. And then we had second oxidation in beta oxidation, and here we have second reduction. Yes? For the reduction, you need an ADPH. Here you only remove water. No, the enzymes only to know that it's reductase, dehydratase, and reductase. Yes? This we can skip. Here we have the reduction. The first reduction, you have acetoacetyl bound to acyl carrier protein. And in the first step, you reduce this keto group to hydroxy group. And you need an ADPH. In the second reduction, you remove this hydroxy group plus this hydrogen. You release water. And here you produce the double bond. Yes? In beta oxidation, it was reverse. On this double bond, you add water. And now you have to reduce this double bond. In the second reduction, so this double bond is reduced by an ADPH, and you produce the linear fatty acid with 4 carbon atoms. And now you take this molecule with 4 carbon atoms, and you add new malonyl coenzyme A to it. Yes? In this step, you remove 1 carbon dioxide, so you make 6 carbon fatty acid. So you understand it's a cyclic process with many rotations, with 7 rotations, and in every step you add 2 carbon atoms. And then you do reduction, removal of water, and reduction. And the new malonyl coenzyme A. Is it clear? Yes? The only problem that you will see in the books is that the intermediates change their position between acyl carrier protein and condensing enzyme. Yes? It's not necessary to understand all the steps. Yes? Understand the first step, how you form the malonyl coenzyme A, then the condensation reaction, and then the first reduction, removal of water, and second reduction. Yes? You end when you have palmitate, it means 16 carbon atoms. Yes? When you have 16 carbon atoms, the palmitate is removed from fatty acid synthase by the enzyme thioesterase. It removes the palmitate from the bond on acyl carrier protein. But you know that in our body we have many other fatty acids. So we have to have other ways how to produce them. Yes? Do you have questions or not? Is it clear? For you, sometimes look that it's not clear. Good. So when it's clear, then before the break, I will tell you that to produce 1 palmitate, we need 3 compounds. We need acetyl coenzyme A, we need ATP to do the carboxylation of acetyl coenzyme A to malonyl, and then we need 14 NADPH molecules. Yes? The ATP is necessary to do the carboxylation, and ADPH is necessary to do the reduction. Yes? And we will gain these compounds from different ways. Acetyl coenzyme A we will gain from pyruvate dehydrogenase reaction, that's mitochondria. And an ADPH we will gain from pentose cycle and then from one specific pathway. Yes? And that's after the break. So before the break, we ended with information that for palmitate we need 8 molecules of acetyl coenzyme A, 14 molecules of NADPH, and 7 molecules of ATP. ATP is produced in mitochondria, especially in pyruvate dehydrogenase reaction. Yes, when we convert pyruvate to acetyl coenzyme A by oxidative decarboxylation. Now we need to transfer this acetyl coenzyme A to cytosol. yes, where we have the synthesis of fatty acids. The problem is that the inner mitochondrial membrane is impermeable for acetyl coenzyme A, and therefore we have to find some other way how to transport it, and the way is citrate shuttle, yes. This citrate shuttle is very nice shuttle, for this transport is also able to produce 8 molecules of NADPH, and it means that from the pentose cycle we need only 6 molecules to gain these 14 molecules, yes? So the function of citrate shuttle is not only to transfer acetyl coenzyme A from matrix to cytosol, but also to produce the NADPH. And here you have the scheme of the shuttle. I think that you didn't spoke about it during the Krebs cycle lecture, or yes? For sometimes they speak about it. But what's the problem here is this acetyl coenzyme A that cannot go through the inner membrane. For acetyl coenzyme S is a big molecule, yes? And unfortunately, you also do not use the carnitine shuttle, yes? You use the citrate shuttle. It means that you let react acetyl coenzyme A with oxaloacetate, and you form citrate, the first reaction of Krebs cycle. This citrate has a transporter in the inner mitochondrial membrane, and it can be transported to the cytosol. Here, the citrate is degraded back to acetyl coenzyme A and to oxaloacetate, yes? This enzyme you have to know is the ATP citrate lyase. Here you need ATP. This acetyl coenzyme A moves to the synthesis of fatty acids, yes? But now we need to transport the oxaloacetate back to the matrix, and also to gain 1 molecule of NADPH. Oxaloacetate is in the first reaction reduced to malate, yes? The enzyme is malate dehydrogenase. You know that malate dehydrogenase is also in Krebs cycle. So this is isoenzyme, and we call it cytosolic malate dehydrogenase, yes? It uses NADH as a cofactor, yes? Here we have keto group, here we have hydroxy group. Still the same number of atoms. And then the unique reaction for cytosol is that we can convert here malate to pyruvate. Malate has 4 carbon atoms, pyruvate has 3 carbon atoms. It means you have to remove 1 carbon atom, and you have here carbon dioxide. It means that this reaction is decarboxylation. And also, you gain here 1 molecule of NADPH, so it's oxidation. oxidation. And if in one reaction we have oxidation and decarboxylation, we call it oxidative decarboxylation, yes? So it's the same mechanism as for pyruvate dehydrogenase reaction. Oxidation plus decarboxylation. The name of the enzyme is Melic enzyme. The systematic name is NADP malate enzyme. And here you gain pyruvate. And for pyruvate, you have transporter that can transport pyruvate back to the matrix. And here, to close the shuttle, you have pyruvate carboxylase, the first enzyme of gluconeogenesis. And you add carbon dioxide on the molecule, you consume ATP, and from 3 carbon atom compound, you gain oxaloacetate with 4 carbon atoms. And you can start from the beginning. So this is the citrate shuttle, the way how we generate 8 molecules of NADPH per 1 palmitate, and also the way how we transport acetyl coenzyme A from matrix to the cytosol. So this shuttle you have to learn, the enzymes you have to know. Here you have only 2 new enzymes. This enzyme is new for you, and this enzyme is new for you. Yes? The other you already know. You understand how it works, this shuttle? So, here you have written the things I said right now. Yes, so I will skip it. Regulation of fatty acid synthesis. When we can do this reaction, we need to have energy and we need to have substrates. If you want to produce new fatty acid, the substrates are other nutrients. It means saccharides or amino acids. Yes? You have to degrade them to acetyl coenzyme A. Yes? There's the substrate, and then during the degradation of the other substrate, of the other nutrients, you gain also energy. And here, if you have the substrates and energy, then there is a regulatory enzyme, the first enzyme of synthesis of fatty acids, and that's the acetyl coenzyme A carboxylase that produces malonyl coenzyme A. And there are 5 factors that regulate its activity. So we will start here. AMP inhibits. It means if you do not have energy, the acetyl coenzyme A is inhibited. That's easy. Citrate activates this pathway. Citrate is the message that you have enough acetyl coenzyme A in the matrix. Yes? Therefore, the citrate can activate the enzyme. Then we have here 2 hormones. Synthesis of fatty acids, that's the anabolic process, and therefore it has to be activated by insulin and inhibited by counter-regulatory hormones. Yes? They modulate this activity by the phosphorylation or dephosphorylation. Yes? The same as the glycogen metabolism. And here, the unique inhibition, acetyl coenzyme A carboxylase is inhibited by palmitoyl coenzyme A or by palmitate. For this molecule, that's the product of the synthesis. And if you have excess of this molecule, there is problem somewhere in the cell, and therefore you don't want to produce new. So we call this mechanism as the feedback inhibition. The product of the pathway inhibits the first reaction or the regulatory reaction in general. Questions? In our body, we have many other fatty acids, not only palmitate. We have fatty acids that are longer. We have fatty acids that have double bonds. So how to produce them? The process how to produce double bond or how to prolong the chain is quite complicated, yes? These processes aren't localized in cytosol. They are localized on the membranes of smooth endoplasmic reticulum. For example, for the desaturation, you need 3 specific enzymes, yes? And you do not have to learn these processes in detail, yes? Only know that enzymes that catalyze elongation are called elongases, and enzymes that catalyze formation of double bonds are called desaturases. But what you have to know is that there is one problem with our desaturases. For we cannot incorporate the double bond further than on the carbon number 9. Yes? It means that we have, for example, stearic acid that's saturated. And from stearic acid that has 18 carbon atoms, we can produce oleic acid. It's unsaturated fatty acid with 18 carbons and the double bond on the carbon number 9. But linoleic acid that has 18 carbon atoms and 2 double bonds, and one on carbon number 9 and the second on carbon number 12, this we cannot synthesize. For our desaturases can only put the double bond to carbon number 9 and not further. And therefore some unsaturated fatty acids are for us essential. Yes? You have to know at least these two, linoleic acid and linolenic acid. Funny thing is that in our metabolism we have also some unsaturated fatty acids that have double bond behind carbon 9 and we these can synthesize. The example is, for example, arachidonic acid. Arachidonic acid has 4 double bonds and one is on carbon number 14. Yes? And with this fatty acid we can synthesize in the metabolism. Can you explain why? Yes, you can make it from this. You can make it from linoleic acid. For at the beginning you incorporate double bond to carbon 7 and to carbon 5. And arachidonic acid has 20 carbon atoms. And then you do the elongation. So from double bond on carbon 12, you have carbon 14. Yes? So there are some acids that have also double bond behind number 9, and we can produce them. But only if we have in our body these essential amino acids. Yes? Of course, that these amino acids are also essential. For example, the arachidonic acid. But not direct essential, but it's potentially essential fatty acids. So we can produce them, but only if we have essential fatty acids. And the last topic is we have fatty acids, but the storage compound is not fatty acid, but triacylglycerols. So how to produce triacylglycerols from fatty acids? In one molecule of triacylglycerol, you have 3 fatty acids. So the easy way how to do this is to take the glycerol, 3 acyl coenzyme A's, and put it together. Yes? Unfortunately, in our metabolism, it works different. The first problem is that you do not use pure glycerol, but you use glycerol 3-phosphate. How to gain the glycerol 3-phosphate is from dihydroxyacetone phosphate, and dihydroxyacetone phosphate is the intermediate of glycolysis. So the glycerol you gain from sugars, from glucose. When you have glycerol 3-phosphate, you let it condensate with 2 fatty acids. In the first step, you produce monoacylglycerol 3-phosphate, And in the second reaction, you produce deacylglycerol 3-phosphate. And this compound is called phosphatidic acid. Yes? It's the same as deacylglycerol phosphate. Now the problem is that the phosphatidic acid on the third carbon has phosphate still. Therefore, you take the phosphatase that removes the phosphate from the third carbon of glycerol. And in the last reaction, the deacylglycerol reacts with the third fatty acid, and you produce triacylglycerol. And what to do with this triacylglycerol is to transport it to adipose tissue. How it works, you will hear in the summer course. For the transport of triacylglycerols through the body is quite complicated. We use special compounds that are called lipoproteins to do this. Yes? It means in your books, you will read also about the transport of triacylglycerols in blood, about the lipoproteins, about the very low density lipoprotein, high density lipoprotein. But this topic you do not have to learn for this test. Yes? We'll speak about it in summer course. That's it. So.
Do we need to know like all the process from this one?
For this one, I am absolutely not sure whether it has to be the whole in the test, but
More or less.
more or less. You know, this step, of course, you have to know, and it's not very difficult. 2 times fatty acid, then remove phosphate, and third fatty acid. Of course, the enzymes you do not have to know, yes? But approximately how it works, you should know. You know, these intermediates are very similar when you need to synthesize. When you need to synthesize phospholipids, yes. So I think that you should know, especially what is the phosphatidic acid, for example. Yes. This pathway is quite easy. Against this, the glycolysis, the big mountain. When?
Where?
Where? This also in cytosol.
Of all cells?
That's a good question. For you know that to produce fatty acids, you need energy and you need substrates. So it means that all cells that have these 2 things can do it. Unfortunately, not all cells in our body has energy and substrates. Therefore, the synthesis of fatty acids is especially potent in liver, in skeletal muscle, smaller part, and in adipose tissue. So on the first place is liver, then adipose tissue, and a small amount also in skeletal muscle. Yes? But especially liver and adipose tissue. There you can synthesize fatty acids and therefore also triacylglycerols.
What did you say the phosphatidic acid was the same as?
That this process is similar to the way how we synthesize phospholipids. And that you know from the previous course. So the next week we have the lecture about the regulation. Yes? Those lectures will have 2 parts. The first part, general, how the enzymes can be regulated. So you should remind the 4 lectures about enzymes. And then in the second part of this lecture, we will go through every pathway, and we will repeat the way how they are regulated. Yes? So it's also good to remind this. Yes? It means during this weekend, the whole metabolism.